So what we're going to do is we're going to describe the differences between the appearance of normal and cancer cells. But before we do that, we have to talk about the genetics, because that's the other key important piece in this, is the differences between the genes in cancer and normal cells and why those genes cause those appearances. So the first one we're going to talk about is proto-oncogenes and oncogenes. So before we get to this picture down here, we're going to talk about proto-oncogenes. So during the cell cycle, there's a bunch of checkpoints. There's the growth 1, so G1 phase, and then there's the where the DNA is copied, and then there's another growth phase, and then there's mitosis. Alright, and between each point, these lines signify checkpoints. And that's where these proto-oncogenes and later the other ones are going to come in. Proto-oncogenes regulates this cycle and causes mitosis to occur. So every cell has a different program. Our blood cells live about 120 days, so we have to program this for new blood cells constantly be making, but to fill out so they're replaced every 120 days. Nerve cells almost or never go through mitosis. All our cells are programmed differently. So proto-oncogenes tells this to happen. So proto causes this to happen, but it causes it in a controlled way. So it's a program, all right? So think about a program that's just always going to happen. So if you think about, you set your alarm clock. This is like an alarm clock being set up at 8 o'clock every day. All right, so it tells the cells to divide at that's just an example, but this will tell the cells to divide at 8 o'clock every single day. All right, now what problem is, and remember, cancer is this uncontrollable cell cycle, this uncontrollable cell growth that leads to too much of this. All right, that's pretty much all that cancer is. I mean, it gets more deep than that, but the real basics of it is that there's too much mitosis. All right, so basically what's happening is we have our alarm is going off too many times. So it's like you click the snooze button and then your alarm goes off again, and it tells your cells to go off, and then the alarm goes off again. All right, that is when this is mutated, and it's basically causing too many signals for the cell to divide, and the cell starts dividing a lot more. All right, so what's it called when it divides, and what's the various signals is what we're going to be talking about next here. All right, let's all right, so here we have the proto-oncogenes, and it will when it mutates, we call them oncogenes. All right, so oncology is a study of cancer. Oncogenes causes cancer, and there's various ways this can happen. So if we look over here, we have what happens is, for some reason, there's a new promoter. Promoters cause genes to be expressed. So this causes this to be transcribed into mRNA and make the protein. All right, so now there's too many proteins, so this causes too many signals. Alright, so the cell is going to divide uncontrollably. Gene amplification. So now, for some reason, this gene got copied. And there's a copy, and there's a copy. So now, there's 3 of them instead of 1. So when the cell goes to make the proteins, it's going to make 3 proteins for every 1. So it's going to happen twice as fast. So it's going to get 3 alarms instead of just 1 alarm. And so, again, the cell's going to divide too fast. Alright, now these are mutations pretty much just to the proto-oncogene, but then there's also mutations that cause oncogenes. One is where there's a point mutation. A point mutation is if we have the DNA ATCAGA. A point mutation is basically the same as a single nucleotide polymorph. All right, this one nucleotide right here changed, and now that causes this promoter here to cause this to constantly be copied. All right, so instead of adding, it's basically the same as this one right here. All right, we have this being copied too much, so there's too many proteins being made. Now this one's a little bit different. This one, there's another point mutation, but this is a different type of mutation. This is a mutation within the gene again, and it causes the protein to be it causes one protein, but this protein is different, kind of like in sickle cell. So it's a different protein, and it causes too many signals. All right, it's hyper. It causes way too many signals. So instead of one of these proteins, this one causing one signal, this one will cause maybe 5 signals to divide. And then, or this one, degradation resistant, that means the protein doesn't break down, so it keeps causing those signals. So while this isn't going to be a main focus here, I just wanted you to understand that if you didn't already. But the key thing is proto-oncogenes turn into oncogenes and causes too many cell divisions. All right, so if we go back to this mitosis is just going to happen over and over and over again. All right, until it just the cell. So instead of one cell going into 2, and then these cells slowly dividing into 2, you would have your normal cells here. An oncogene will cause these guys to go like super fast, and then you get all these bunches of cells down here. All right, so cells are usually, and that forms a tumor. A tumor is just a ball of cells. All right, so our next point here. And I gotta erase this here. So our next one is tumor suppressor genes. All right, tumor suppressor are the second gene. So the proto-oncogenes are like the gas pedal. The tumor suppressor are the brakes. So these guys can stop cell division. All right, so they can do this 3 ways. The first one is they can just stop the cell division. They can stop the cell activity right here. All right, so P53 is one of the proteins, one of the example of tumor suppressors, probably the most common, but there's various ones. All right, and it can stop the cell activity. All right, so if proto-oncogenes turn into oncogenes, this is like the second step, this guy can still stop that mutation or stop the cell from dividing. If that doesn't happen, it can also try to repair the gene. So at one of those checkpoints over there, it can actually repair the gene that is mutated. All right, before, and then the cells are going to be repaired, and we don't have no cancer. The third thing, if these 2 things fail, it can cause apoptosis, and this is just cell death. All right, so it causes the cell to basically go into self-destruct mode and kill itself. All right, if there's a mutation in tumor suppressor genes, and these 3 things cannot happen, along with the oncogenes. So cancer is not just one mutation, it's a step of several mutations, because several of these are going to have to be mutated, several of the oncogenes are going to be mutated in order for this to happen. But once that all falls into place, once these happen and you have oncogenes, then the cells will divide uncontrollably, because basically the tumor suppressor genes are responsible for these checkpoints. All right, and these checkpoints are no longer there, so the cell just keeps going and going and going and going all around in a circle until you have this, too many cells. All right, so both oncogenes and tumor suppressor genes have to occur in order for cancer to occur. All right, so you need to understand those, because you're not going to understand the next 2 sections if you don't understand those 2 cells. All right, our next thing here. Whoop. is metastasis. All right, metastasis is cancer. So basically, cancer can be malignant or it can be benign, and benign isn't even really cancer. Um, Benign is just a tumor, meaning it's not going to happen. So benign is just a tumor, and we sometimes it can cause problems, but generally it's just going to sit there and it's not a big deal. Malignant, this is cancer. So malignant is the tumor can spread, and that's when cancer starts to be a problem. If it's localized, we can treat it pretty easily. Depending on where it is, we can treat it pretty easily, and we can get rid of it. But what this is a big problem. So this is why you need to get screened, you need to get tested, and you need to make sure you catch cancer before it goes into this, because once it goes into this, then you're going to have serious problems. So here we have the tumor cells, and what happens is the tumor cells actually ask to be fed by these blood vessels, so it produces capillaries to come up into it, because there's so many cells they need more glucose and oxygen to be fed. What happens is then is this tumor starts to break off and go into the bloodstream then through those capillaries that it caused to build, and it starts spreading. So this might be, let's say this is just skin cancer. Skin cancer is not that big a deal if you get cancer in your skin. I shouldn't say that, but it's not going to cause major problems. Where it becomes problems is once it gets into those blood vessels, which there's tons of blood vessels in your skin, then it can spread to, let's say, your brain, and that's going to be a big issue. All right, it's very hard to operate brain, pancreatic, very hard to operate on those. And then it's going to then form a secondary tumor. And what happens when cancer gets really bad, it spreads to various parts of the body, and that's when your outcome isn't going to look too good. All right, so we want to catch the cell at this stage right here before it starts to do any of that. All right, so metastasis is just the spreading of cancer. And now look at our difference between our normal and cancer cells. I think this part is pretty easy. Cancer cells, if you know that cancer cells are mutated and that they divide too uncontrollably, meaning they grow too much, they divide too much, everything else like that, it should be pretty obvious of what is going on. So here's our normal versus cancer cells. So one, they're larger, all right, and see how big the nucleus is in here compared to this one. All right, so they're larger because that growth cycle keeps going. So not only do they divide, but they also grow a lot bigger. You have many dividing cells because they divide too much. All right, so you have all these cells going through mitosis here, whereas these are not going through mitosis. You have variations in sizes and shapes. Again, because that growth is happening too much, we cause various weird shapes in our cells, and they lose normal features, and then they also grow on top of each other. So you can kind of see that in all these. They grow very close to each other, especially in this one because they lack contact inhibition, and they start just piling and piling and piling on top of each other. All right, so that is the review for this section.